Okay, so welcome everyone again to the Green Era Hub webinar series. My name is Barry Crosby, and I'm one of the communication officers for the Green Era Hub project. If you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU era nets, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs in October 2023, and we've had 5 very successful series so far, focusing on circularity in agriculture, biotechnology to advance sustainable solutions for bioeconomy, sustainable food systems, precision agriculture technologies for a sustainable future, and plant health and protection. If you're interested in listening to the talks related to any of the topics from these series just mentioned, our recordings are now available to view on our Green Era Hub YouTube channel. Now, today we're going to continue with Series 6 of our Breakfast Club webinar series, focusing on the topic of animal health and welfare, which features expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. And just a few housekeeping announcements. Please do feel free to type your question into the Q&A and chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have this morning, which is 1 hour. If at the end of the session you have questions for either of our speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Professor Anne Lespine and Professor Hans Nauwynck, and we will open our session this morning with Anne Lespine, Director of Research at INRAE in France. Based in Toulouse, Professor Lespine has more than 2 decades of experience advancing the understanding of anthelmintic drug mechanisms, resistance evolution, and innovative strategies for parasite control. Her research focuses on the molecular and pharmacological basis of anthelmintic resistance in parasitic nematodes, with the goal of developing sustainable and effective solutions to safeguard animal health and productivity. Professor Lespine has led and contributed to numerous national and international research projects, collaborating closely with veterinarians, industry partners, and academic institutions to translate scientific findings into practical tools for livestock management. She's widely recognized for her contributions to the field of pharmacology, parasitology, and veterinary sciences, and her work continues to shape strategies that help farmers and veterinarians respond to the growing global challenge of drug resistance in livestock parasites. Today, Professor Lespine is going to be speaking about her research project entitled Haemonchus Anthelmintic Resistance, Evolution, Mechanisms and Innovative Solutions, which is part of the EU-funded International Coordination on Research on Infectious Animal Diseases, the ICRAD initiative. And thank you very much for accepting our invitation to the webinar series. We're absolutely delighted to have you join us this morning. I'm going to stop sharing my screen now, and you can start sharing your presentation. Thank you very much.
Thank you, all of you, for being here, listening. So the objective today is to present one of the ICRAD project that we submitted last year and has been accepted for financial support. It's called Artemis for Haemonchus Anthelmintic Resistance, and we are supposed to study the evolution mechanism and innovative solution for the problem. So let me please introduce you our work. I'm working in a unit of INRA. INRA is a research institute, national research institute with agronomic interest. We work in our unit of 60 persons on a veterinary drug of clinical importance, such as antibiotic, and my main job is antiparasitic drugs, such as anthelmintic or insecticide. And the point is, these drugs are highly used, massively used to treat infection disease in animals, vet, like pets or farming animals, but also in humans. They are efficient against a lot of targets, parasites, insects, or nematodes. And the problem today is that there is drug resistance everywhere. It's a little bit like antibiotic resistance, but concerning other drugs. So we are supposed to work on that and to solve the problem. The context of Artemis program joined this interest. It's animal health, parasitic infections, and we are focusing on gastrointestinal nematodes that affect animal welfare and health. This is a worldwide problem affecting the economy of animal production. We focus on certain drugs which are macrocyclic lactones because they are the main used today. They are obligatory to control gastrointestinal nematodes. And today, the fact is that there is resistance to all anthelmintics and a very slow development for new drugs. So it is in this context that we are all working in my unit research. The Artemis objectives are more focused on a specific parasite, which is Haemonchus contortus. It's a gastrointestinal worm, very pathogenic for small ruminants. He has a high genetic variability, which make him very adaptive to all the stress. And that's a real problem. It has a high ability to develop in some situations such as high temperature, drug exposure. It can adapt. We are supposed to study this worm from the livestock to the genomic, and that's the challenge actually we have here. We want to study him at the regional level, European level, and look at the impact on farm economy. So the project is very, very broad, and that was a real challenge. The overall objective, so it's investigate into spreading of resistance to anthelmintic and Haemonchus contortus by identifying molecular mechanism of ML resistance, extending to our research to Europe, and exploring climate impact and economic cost. And this means that we will go to the farms, to the lab, and come back to the farms with solutions, hopefully. The consortium partners cover France, of course, because I am the leader and we are two laboratory working on that. The one is my laboratory in Dres, and the other one is the veterinary school with involving vet, which are a very important part in this project. The other partner is UK, the University of Belfast, with Eric Morgan, which is specialist in the field of epidemiology, including anthelmintic resistance in parasites, and Poland, with the laboratory of Marcin Mikiewicz, which is a leader in epidemiological studies and economic impact on gastrointestinal nematodes. The operational objectives of the program will be to track the evolution of the parasite populations and of the pharmacoresistance in the context of climate change in UK, Poland, and France. This will provide knowledge in C. elegans and Haemonchus contortus on the mechanism of drug and temperature adaptation and predicting efficient drug when one drug become inefficient. Okay, I add C. elegans, which is a very good model of nematodes, and it's easier to use it in the laboratory and to understand molecular mechanism. The strategies to control infections in farms and to evaluate economic costs and benefits of these protocols. So the organization is provided in several work packages. The fifth work package is a coordination, which is made in my lab by myself and my team. The work package one is led by University of Belfast, Eric Morgan, and the objective is to follow the evolution of parasite community and anthelmintic resistance in the three countries, France, Poland, and UK. The other work package is to understand the molecular components of adaptation in nematodes, C. elegans and Haemonchus contortus, and this is led by our laboratory in France, in Toulouse, Interest Laboratory. The work package 3 is implementation of alternative solution to anthelmintic resistance in farms. Once there is resistance in the farms, we go there and propose solutions to avoid use of chemical as much as we can. And this is led by veterinary school in Toulouse, in France. And the work package 4 is an economic component of the project and socio-economic with the impact of Haemonchus contortus infection on the cost in the farms. And this is led by Poland, University of Varsovie. So this is the global issue of the project Artemis. And to illustrate what we're expecting as data, I would like to show the first data we have in our regional context. And this is the support that we want now to expand to European countries involved in the project. So the French context with who we started with, the story started in France, in the southwest of France, where there is a high domain of cheese production, when flocks are infected with Haemonchus contortus, severely infected. We use one specific drug, which is a predomectin. It's class of macrocyclic lactone, and it's the last anthelmintic authorized in dairy farms without milk withdrawal period. And this is a very important component. However, we are now confronted to epinomectin resistance. And that's the problem. So we started a previous survey and study financed by a local finances in the Pyrenees-Atlantic, which is southwest of France. It's a mountain area, and we identify several farms, with some of them have lack of efficacy in the fields. So this is 6 farms, very closely related. And we went to this farm and established a protocol that we want to proceed also at the European level. We went to the farms, treat the animals, and do a fecal egg count reduction test. This is to follow the efficacy of the drug. We treat with epinomectin and collect the feces, count the eggs. That's typically how it's performed. And beside, we measure the drug in the plasma of the animal to be sure that the drug is well there and could be efficient at the level we are expecting to be efficient. So once we have done that, very sensitive, sorry for that. We phenotype the worms in the lab by evaluating their motility in presence of drug. When the drug is there, the motility should decrease. If the worm is susceptible, and here at the right-hand side, you can see the curve that represent the evolution of motility in presence of drug. Green is when they are susceptible. The curve is to the left, sorry. You don't need a lot of drug to be efficient. And red, they are resistant. And this is actually what we obtain for the lab. Oh, sorry. The other step is to take the worms, isolate the DNA, RNA to make a transcriptome, to evaluate the transcriptome and the genome. And we sequence all of that, and we perform a data analysis. And this is what I'm going to present, that the first data we obtain from very localized farm in France, and we wish to obtain in other countries also. Oh, sorry. So this is the first table which present actually the isolates and the farms on the left. This is the name of the France, all collected in Pyrenees Atlantic, and this is a date, and the peak concentration that you see here are consistent with the efficacy expected. And when we look at the FECRT, which is the fecal count reduction test, we obtain limited results. We are expecting between 95 and 100% in the susceptible strain, which are here 100%, and this is a susceptible strain in blue. The other forms in red are expected to be resistant because the fecal count reduction tests show very low values, okay, compatible with the resistance status. And to verify their resistance, we conducted our in vitro test here, following the motility of the worms in presence of the drug. The two green left curve are the susceptible one with the EC50 very low, and the four red curves corresponding to very high resistance factor. They are resistant to eprenomectin. This is consistent to what was obtained at the farm level, and this is in the lab, actually. So we went further into the genomic of these worms. We took larvae, explored the DNA, and compare actually two susceptible strains here, and this is a genetic variation, genetic differentiation all along the chromosomes. Each point are a region on the chromosome, and they are, when you compare two susceptible strains, you don't see very high variations, at least statistical variation, differentiation. But when we compare one resistant with one susceptible, then here you can see clearly some regions that we will call QTL that are highly variable, or highly differentiated at least. One is on the chromosome 5 here, and this is interesting to remind that this region was also shown on the resistant Haemonchus contortus resistant to ivermectin, which is another macrocyclic lactone. This is eprenomectin resistant strains we are looking at. And this is the first evidence of a change of the genome of eprenomectin resistant Haemonchus contortus. This is a first view, actually, that nobody has explored the genetic variation so far. The other point also interesting is the variation, the differentiation on the chromosome 4 here. This has never been observed before, even on ivermectin resistant strains. Means that this is maybe specific of Haemonchus contortus eprenomectin resistant. So that's two very new information on our strains collected in France. The other information here concerns the deregulated gene, and this needs transcriptomic. We sequence the RNA and look at the differentiation deregulated gene in the high, upregulated, which is yellow, and downregulated, which is blue. And here we can see that there is some nice variation, clear variation, statistically correlated in male and in females on the chromosome 5s, okay? And there is some variation also on the chromosome 4, which sounds to be specific on female. What was interesting to focus on here and to notice is that this variation corresponds very clearly to the variation observed on the genetic differentiation. So this is RNA, deregulated gene, up and down regulated, and RNA differentiation with mutations. Okay, so you have regions here that are highly important in the resistance to eprenomectin. This is a very, very important information and very new, but this is very localized in our isolates, in our regions. So the expected outcomes of Artemis is to improve the understanding of anthelmintic resistance, eprenomectin resistance in European countries, in Haemonchus contortus, from urban, large, broad urban countries, to develop diagnostic tools to predict the evolution of infective parasites and to counter the spread of anthelmintic resistance, and to propose achievable strategies to address the problem of anthelmintic resistance in farms. This is also to anticipate the impact of climate change, and this is by looking at different regions of Europe, the UK, Poland, and France. But by identifying molecular targets, as we did actually, and to come back to the full proposal is first, go into the flock, identifying climate change, temperature, hygrometry, chemical stress, combining with chemical stress. Go to the worms and to identify genes, the protein functions. This paved the way for identifying novel targets to improve the diagnostic and the treatments for helminth infections. And towards solution for improve also our animal productions. So I just want to present you my team that participated to the first initial project. And we are continuing now with our colleague in Europe. And of course, thanks to ICRAD for their financial support. Thank you all for listening. And of course, now just I'm here to answer to your questions.
And thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Professor Lespine at any point. And just while we're waiting for one or two questions to come in, if I could just maybe take you back just to the very beginnings of the Artemis project and the kind of broader aims and objectives about the project. And I was wondering
Can
if
I
you
come
can
back
tell,
with the slides?
yeah, of course. I was just wondering if you can tell us maybe a little bit about the parasite itself, a little bit more about that, and particularly what makes Haemonchus contortus such a particularly challenging parasite in terms of developing and spreading drug resistance.
Okay, this parasite actually is was specific from a tropical country in France. We were studying it because it was in our dom-tom, Guadeloupe, or all the tropical dom-tom. So it was a very interesting point. Most of the gastrointestinal parasite may slow down the production or limit the production, but not so much. And the animal can live with it. Haemonchus contortus is very pathogenic and reproduce very rapidly. The load in the animal can be very high, is suckling the blood, so there is anemia and diarrhea, and the animal can quickly die from this parasite, which is not obviously the case for the parasite. So it's dangerous. But it was constrained to a tropical country. However, since recently, 20 years, we find it was found in our mountains, but with the winter, it disappeared. Now it stays all the seasons and can survive, which is a new thing for us. And this is linked to the obvious temperature changing. The winter are not so cold, and this parasite can survive. And it's found also in northern country like UK, Poland, where usually he was there, but not very, very important. Now it's an important issue. So we need to, he reproduces very quickly, and he has a very, very high genetic variability. Means that when you expose it to something, it will try in a way to adapt genetically. And this is what happened with drugs. Also with temperature. So it will change its kind of fitness and everything in order to survive.
suit the environment.
which
That,
make it very, very, how do you say, skin. We need
yes.
to fight against something which is always changing. And so this is why genetically it's very important to be able to see changes. And we are betting now that we will not see the same changes in UK or in Poland. Or maybe yes, we don't know, but that's by brooding we will have more impact in our research.
And sorry, just on that note, how do you think how does resistance in Haemonchus compare to, for example, what's observed in other gastrointestinal nematodes in livestock?
They may share some aspects of resistance. The problem is maybe not the nematodes, the target nematodes, it's the drug itself. Macrocyclic lactones are complex drug. I mean, the resistance mechanisms are not those which were expected. In terms of pharmacology, one drug actually provides resistance by inducing a mutation in the target gene. This was the case for previous very efficient anthelmintics, which were benzimidazoles. Benzimidazoles are still used in humans, but in vet, they are not efficient at all because one mutation appeared on the target of the drug, which are beta tubulin. And one single mutations changed the interaction of the drug with the target. And that's enough. And this is called target resistance. And once this show up, it's transmitted to other generation. And we cannot use any more benzimidazole. This is why macrocyclic lactones have been developed in the 80s. And we have been using them very efficiently until recently. Because the process of developing resistance is more complex, more long, and now we are facing resistance. But when we're looking at the target genes, there's no mutations on the target gene of macrocyclic lactone. It's a polygenic mechanism, and we don't know actually what are the target and what are the mutations that will drive directly to resistance. We are looking at QTRs with a lot of genes inside. We don't know which ones are important. This is why, and this is an important point of the project, beside Haemonchus, we run also C. elegans. C. elegans is an organism that we can run in vitro in our lab, and we can have very, very rapid answers to the targets. And this answer partly to your question is are the other nematodes predictive of what would appear in Haemonchus. Some of our targets we found in C. elegans are also found in Haemonchus, and that's helped a lot. However, not all, like some are specific of Haemonchus, so this complexifies the problem. But gathering everything together, isolates that we will collect of Haemonchus that we will collect in the farms, and C. elegans that we have in the lab will certainly provide a lot of answers, and certainly definitive answer, hopefully, we hope.
That's fascinating stuff and fascinating. Just a question from Emily in the audience, just in relation to transmission. Is the parasite transmitted through food?
Yes. When a sheep is parasite, this is adult swarms which are supposed to be in the stomach of the animal, and he will release eggs. Eggs will be released in the pastures, and they will develop into egg-free, and the egg-free is one egg-free that will land on the grass in the water droplets in the grass, waiting for the animal to eat the grass, and the L3 will enter the stomach and develop into L4 and adult. And the cycle is there. So if you have a flock with one animal which is contaminated, it will contaminate all the other one. And if you exchange flocks, and that's one of the part of our program in France, pastoral system, which is a very nice system to produce very nice milk and very nice cheese. We like cheese from pastoral system in the mountain. It's element we are publishing, recently we published that favor the exchange of resistant parasites, because the flock exchange in the pasture in the mountain, flock from one farm to another. So if one farm is parasitized by resistant parasite, it will exchange with the other one, which is a kind of problem we were not aware so far. Now we are aware, and we can limit this exchange. And that's, more we understand, more we will be able to act on the problem. That's the message of that. So the cycle is clear. You understand it clearly.
And thank you so much for that wonderful view of your project. I think it's a fantastic project, and I think we could be here easily for another half an hour asking questions. But thank you very much. We'll just have to move on to our next speaker now, unfortunately. But thank you very much for Professor Lespine for joining us today and for giving us what was really an excellent presentation.
Thank you, Barry. Thank you
Thank you
all
very
for
much,
being
Anne.
here.
Thank you. So now joining us is Professor Hans Nauwynck, Director of the Laboratory of Virology at the Faculty of Veterinary Medicine at Ghent University in Belgium. Professor Nauwynck graduated as a Doctor of Veterinary Medicine before obtaining his PhD in Veterinary Sciences in 1993. Since 2004, he has led the Laboratory of Virology and is a diplomate of the European College of Pig Health Management. His research focuses on the cellular and molecular pathogenesis of viral diseases in humans and animals, with particular emphasis on how viruses enter primary target cells, cross host barriers, spread via leukocytes and neurons, and escape immune responses. This work drives the development of next generation treatments and vaccines. Professor Nauwynck has authored more than 400 scientific papers, supervised over 70 PhD students, and is the inventor of multiple patents. With a team of more than 35 researchers and technicians, he continues to be at the forefront of veterinary virology and is a frequent keynote speaker at international scientific congresses. Today, Professor Nauwynck's talk is entitled Use of Frontline Technologies to Screen Pathogens, Environment and Pigs for a Better Disease Control in Swine Herds, which is also part of the EU-funded ICRAD initiative. Hans, thank you very much once again for accepting our invitation to our webinar series. We're absolutely delighted and privileged to have you joining us this morning. Without further ado, I think I'll just open the floor to you. I'm going to stop sharing my screen and you can start sharing your presentation. Thank you very much.
Good morning, everybody. First of all, I would like to thank Barry for giving me the floor and present you some data, some results that we had during a project, an ICRAD project. It was called TechPepCon, and the long title was Use of Frontline Technologies to Screen Pathogens, Environment and Pigs for a Better Disease Control in Swine Herds. And this was a consortium, and a consortium consisting of 5 countries. That was Belgium, Poland, Greece, Hungary, and Italy. In the beginning, we had also Russia, but due to the circumstances, the war circumstances, we were not able anymore to cope with the Russian partner, which was of course which was a pity. But okay, it is what it is. And what I would want to do is to take you in let's say the problem that we had and how we wanted to solve this problem. And first of all, let's have a look. Go to the next slide. Yeah. So I will use my pointer if possible. Yes, here, laser pointer. Okay, here we go. And everybody of you know that there are a lot of problems on pig farms, and the biggest problem is how to control diseases. And everybody knows also at this moment that those diseases are not single etiology diseases; they're always a complex. And if you want to approach a complex to control, then that is also complex. So, and what we wanted to do is, and that's the start of everything, of course, how to make a clear diagnosis. What is the cause of the disease? And especially not the single cause, which is most veterinarians want to have the cause, but this is not logical because it's mostly a complex that we have to identify and then take care of it. First of all, what did we have in the past? Well, in the past, if you're a vet and you go to a diseased herd, then you have, first of all, an anamnesis. That means you talk with the farmer. First of all, what are we dealing with? What is the evolution? Evolution on the farm, evolution in the animals. Very important also is to discuss about the biosecurity. So this takes quite some time to have a clear idea on what's going on the farm. Then the next thing you do as a vet is you grab some pigs or you look to the pigs and you make a kind of a clinical examination. And very important is on the right, you can see here an analysis of the environment. So as a vet, you have to run around, you have to look, you have to see, but look to the temperature, you have to smell also. So you should use all your senses to have an idea on the environment. Also dust, if you look to the, especially to the feed of the animal and the environment. So these things you should all take care of, which is not easy because our senses are good, but they're not extremely good. So we have some restrictions concerning our senses. Then afterwards, what you do is if you have some ideas, because they call it the differential diagnosis, then you have to sample the living animals. And also, if possible, sample some dead animals. And very important is then you do a sampling. So you take some material and you send those samples to the laboratory for laboratory analysis. And very important, in the good old days, what did you do is you were ordering tests. So you wanted to identify a certain virus or a certain bacteria, and you did that with PCR. So if you have a PCR, then you use primers, of course, because for every virus you need specific primers. And then you were looking specifically for a certain virus or a certain bacteria. You could also use ELISA, or in the good old days, immunofluorescence or immunochemistry. And then, of course, you need antibodies to identify your virus or your viral antigens. And very important is the good old days, that was virus isolation. For virus isolation, of course, you needed some cells or embryonated eggs. So you really always had to have a kind of an idea of what you wanted to test for. And this is, of course, giving a lot of restrictions. You only get the things out what you're looking for. But as I already told you, the disease is most of the time is a complex. So you could make a wrong choice at that moment. So these are, this is then what you have to do as a vet. You put all those things together in your brain, and then you start to have an idea on what the diagnosis was, and then you could control the different parts that were important. Then, as you can see on the right here, if you have all those data, I can tell you this is not easy to put all those things together in your poor brains. We have limited power in our brains, and that was the starting of this project. We wanted to use new technologies and integrate them and make it more easy for the vet to put all the things together and come up with a diagnosis. So this was the old way of doing the things. So if you look now to how we could change this by using new technologies, first of all, if you look to the problem, we would like to see numbers on the screen, which is possible nowadays. We should also see we should have an idea on the biosecurity, and this is possible. We have here at the UGent a spin-off called biocheck.UGent. If you're interested, you can go there. And that's a questionnaire. You have to fill in the questionnaire as a vet or a farmer, and you can end up with scores. Scores about the internal biosecurity and also the external security. So this is extremely important. You get a score and you can deal with that. So this is all data that comes on the screen of your computer. Clinical analysis, of course, up till now, that's still in the hands of a vet visiting the farm and looking to the animals. However, we have here, and on the right you see here a very important apparatus. This is, they call it a healthy climate sensor. And on the sensor, as you can see here, you go for several, you can do a lot of things. So first of all, it's smelling gases, concentration, CO2, ammonia. It's also identifying dust particles at different sizes. You get there an outprint. It's also detecting the temperature on the farm. And there's also a camera and also a microphone, but I put this already in between brackets because these things, we used it, but it was not very functional. So we still have to work on these two. And this is important because this will help the vet. So if you have a vet going into a farm, this vet has to look into farm, but he can also use a camera and also a microphone to have a better idea what the problem is on that farm. But that's, you know, we still need to have some development on those two parts. Then, of course, in the good old days, if we were selecting material, then we send it to laboratories for PCRs, ELISAs, and so on. The new approach is another approach. So when you sample animals, living animals, or at necropsy, you can do, and we call it third generation sequencing on the sample. This is easy at this moment because you can see here a Minion. That's a little machine, not the Minion. That's another thing. It's the Minion, and this is a very small device. You can see here the computer, and you put your sample on top of that, and then your sample will run through this little machine and will come up with sequences. And the sequences, the reads that you get out of that, you can assemble those reads, and then you can come up with big sequences, and then you can identify viruses, bacteria, and now recently also virulence factors. So you get out of the sample everything without a foreknowledge. So you don't have to select. You just select. You just analyze the whole sample for everything what's in it, and this is working extremely well. This is also a spin-off from UGent that is now working on it itself, and you can see the thing is already available all over Europe. So this is a new technique. No foreground, you don't need to think about, you just sample your diseased animal. If it is respiratory tract problems, you will not take a fecal swab, of course, you will take a nasal swab. If it is a problem at the level of the lungs, you can use tracheobronchial swabs. If you have at necropsy, you can make incisions in, for instance, into the intestines or in the lungs, and you can send it and you can have the full analysis. So this was totally new also. And then for us, very important is then you have all those data. And then the question is, we should have all those data together. And we worked on that in that project. And we came up with I call it the one pager, which is always important to put the things together. So on top, this is a view that you get from the biosecurity levels on the farm. Then this in the middle, you see there environmental parameters, like you have here particles, CO2 concentration, temperature, relative humidity, and at the bottom these are from left to right. This was a period on a farm, so we were following a farm, and these at the bottom were in samples were pathogens that were detected in the samples. And as you can see, there were a lot of pathogens circulating on those farms. So what we did in the project was, first of all, to have an idea on healthy farms. Because you have to start, you should have a kind of a basis, the background. And we took in Belgium 5 Flemish healthy farms. And our expectation was to have no disease, no biosecurity problems, a healthy environment, and no pathogen circulation. That was our idea because they were doing well on those farms. So we started to do that. These are an overview. We will not go in detail. Clinical signs, sneezing, coughing, and diarrhea. And these are the different farms that we had and the time point on top. And you can see, although people said, yeah, we have no problems, you were hearing a lot of sneezing, coughing, and there was also diarrhea. So we already had some problems with saying there was no disease on those healthy farms. If you went into those farms and you were analyzing it carefully, we saw some clinical signs. Then the biosecurity, this is the way we do it. This is presented. So the bold line here in black, that's the country average. This is Flanders. And then you have all the farms, they have different colors. And the more they go to the outside, the better. And you can see, for instance, location of the farm. Also here, cleaning has disinfection. Sometimes the farms go underneath the bold line, showing that there were problems with the biosecurity. So also there, even when we talk about healthy farms, there are biosecurity issues. So that's a working point. Then concerning the healthy environment, but just looking to the healthy environment, we saw, for instance, in the farrowing unit on one farm that there was a lot of production of dust, which is not good for the respiratory tract. We also found out that here in when the piglets went to the finishing unit, we saw that the temperature was too low, and related with that the relative humidity was too high. And that also in the finishing unit, you can see it here, we had too high concentration of CO2, so the ventilation was not very perfect. So even there, when we were looking in detail, healthy environment was not very healthy on those farms. And then we were looking to the pathogens, and this is surprise, surprise. So in blue, you have the nasal swabs, in orange, the tracheobronchial swabs, and in gray, the rectal swabs. And as you can see, these are the mean number of viruses on those farms. This is the age of the pig, and you can see there are a lot of pathogens circulating, both respiratory and alimentary virus. So this was for us also an eye-opener to show that there's a lot of circulation, even in environments where you think everything is okay. So even there, we had to scratch that. And that shows that even in Belgium, and we saw that also in the other partners countries, that healthy farms have problems already. And there should be actions improving biosecurity, improving environmental conditions, and also very important, reduce the pathogen circulations. As a virologist, I'm very interested in that because every time when a pig gets infected with a pathogen, this virus or bacteria will start to replicate and will stimulate the immunity. That is called in veterinary term, this is called a lymphoblastogenesis. So you get a lot of lymphoblasts. These are lymphocytes in proliferation, in mitosis, and these are the targets for very small DNA viruses, the porcine parvoviruses and the porcine circoviruses. And those viruses are just exploding at this moment. And you should know that this is not done. We should really try to take care of this, and we should really get the number of pathogens that are circulating, we should get that down. So very important is these were the healthy farms. Of course, if you see already the problems that we have on healthy farms, you can imagine that this gave some problems, because if you get an isolation of a virus or a bacteria on a diseased herd, then you should question yourself, is this pathogen related with the problems we have on that farm? And that makes it at this moment extremely complicated and not easy. And we did we did a lot of affected farms, hundreds of those farms. Of course, you can imagine we cannot go run over all those different farms, but there were many more problems concerning biosecurity, environment, and pathogens, of course. And combining all the data from all these new technologies gave better insights in the etiology and how to control the problems on a farm. So we could really direct our approach to control those problems. However, and as you can see here, this is just a cartoon. This is not one of my co-workers, but the integration was problematic. And very important to mention here is that our brains do not cope anymore with this data explosive evolution. So there's too much data that, we create so many data that our brains, poor brains, cannot cope with it anymore. And there's only one solution in the future that we can, how to deal with this. And this is just to add here artificial intelligence. And we should have at the end help so that we can have a kind of a simply overview of what is going wrong on a farm that will send then the message to our brain so that we can deal with it. So at this moment, this is now, I would call it a nightmare, but I'm convinced within this and 5 to 10 years, I would call it, this will be the new normal in veterinary diagnostics. And this will be thanks to artificial intelligence, because everybody is scared of artificial intelligence. You don't have to be scared. You should use it in a correct way. That's the way of looking to this new technology. So this is what we did in that project. This is not the end. So every single new technology, so the sensors, biocheck, and also the pathosense, all those new technologies are evolving further in a very good direction, and there's a lot of interaction. So we're talking with all those different developers, and at the end, I'm convinced if we put a sauce of artificial intelligence on top of that, that it will become easier for a vet to come to a diagnosis and to come to very, directed control of diseases on farms. So, Barry, this is what I had to tell. I hope that the people are still following. And if there are some questions, I'm happy to deal with those questions and looking forward to those questions. Thank you, Barry, and thank to the audience. Thank you.
Hans, thank you so much. That was a really, really interesting and wonderful presentation. Just want to remind everyone that the chat box and Q&A section is open, so please do feel free if you have a question for Professor Nauwynck at any point between now and the end of the webinar, please do feel free to type your question there. Hans, I was just kind of fascinated just by your kind of description of the main types of kind of challenges that you face in kind of putting together and initiating a project like this. And I was particularly interested about how, complex these diseases are. You mentioned, for example, how difficult they are to detect even on so-called kind of healthy farms. And I was just wondering, for me and for the audience, if you can maybe talk a little bit more about that, about the particular, what you see are the main challenges in current swine disease control that these new screening technologies and indeed AI hopefully will kind of seek to address or aim to address.
Yes, okay, Barry. So in the past, let's say the diseases that we saw on farms were mainly diseases that were caused by a certain pathogen. For instance, Ojeski's disease in the good old days, pseudorabies virus. These symptoms were so clear. You had in the piglets, you had central nervous disorders. In older pigs, you had anorexia and high fever. In the sows, you had abortion. They were very clear. The symptoms were clear, and it was easy to make a diagnosis. There's another virus like a transmissible gastroenteritis virus. That was a coronavirus. That was very easy to diagnose because you had a high mortality. So diarrhea in young piglets. The animals were not protected by the mother. So you had a mortality up to 100%. So these were, I call it, the single etiology diseases, that was very easy. But nowadays, these diseases, there are still a few of them, but most of those diseases are multifactorial and multi-pathogen complexes. So if you see something on a farm, let's say sneezing, for instance, you hear it and there's a lot of sneezing. Then you could say, okay, what could be the reason for that? It could be, of course, cytomegalovirus is linked with that. PRRS can be linked with that. You have also some bacteria like Pasteurella, Bordetella. So you have a lot of pathogens that are involved there. So you cannot say, I will test it for PRRS, and vets do at this moment, and that's totally wrong. You cannot say, I will test it for PRRS and say, okay, I isolate PRRS, and it is the cause of the problem. You cannot do that because PRRS is circulating on every farm and is on most farms not involved in disease. So you have to be very, very careful. That's why you should, if you go for the pathogens, you should have a look in a broad way. And looking in a broad way, that means you should, try to find all the pathogens that are there and are making the complex. And then I remember the diagnosis with pathogens. That was a complex of PRRS, cytomegalo, Pasteurella, and Bordetella in one sample. If you see that is, I call it a textbook of atrophic rhinitis, a textbook case. And you got it all in your sample. And that is very important because you can take care of every single one. So if you say PRRS, you can control PRRS. That's not a problem. Cytomegalo, that's a difficult one. At this moment, there's nothing to do on that one. But for instance, then you have your Bordetella and the Pasteurella. There you can play around with. There are some vaccines also. So with looking to the complex, you can work on every individual one, and you can. I call it destroy the whole complex. And we know also that the environment. So I'm convinced if you went to that farm and you went to have a look to the ventilation, to the dust, you could also see some things that are going wrong there. So with adding all those sensors, all those new technologies together, you could come to a very good approach to control every little component of that complex instead of only hitting one, which is mainly done at this moment. So we really have to have a broader view on controlling diseases than we have at this moment. And if there would be no possibilities, and would say, poor me or poor vet, this is impossible. But the technologies are going extremely fast, helping them a lot. The only thing we have to do is trying to get the things together and translate it for a vet and a farmer in the field. And that is what is lacking at this moment. And the vets at this moment are a little bit reluctant to use the new technologies, because they always say, all that information, what can we do with it? And indeed, our brains cannot cope with that. And we need, I call it an easy translation towards the vet in the form of artificial intelligence that will translate it and make a kind of list of doable control measures that you can take on that farm.
That's fascinating, Hans. And I was just going to ask you actually about that. You just touched on my next question, which was about what you see as the main barriers to adopting these new technologies on commercial pig farms. So is it cost? Is it actually kind of dealing with the technologies and training farmers and veterinarians? What do you see as being the kind of the main obstacle or barrier for the kind of wider embracement of these technologies?
As a disease is caused by complexes, this thing, the basis of the people that are a little bit, have difficulties to use the new technologies, is the first thing is, of course, the education. The training of the new vets should go in a direction that they are more using sensors or new technologies on the farm. And this is an ongoing process, but it's not going fast enough. So I think the education at the universities are playing an extremely important role there. That's one thing. A second thing is also the price. There's always a price, and nowadays the farmers are suffering. Everything is low prices. So if you're not paid well as a farmer, you will not invest in proper diagnostics, and that's also a problem there. Then also putting the things together, because they're sometimes scared to have the full insights in the complex. Because, for instance, you have 3 or 4 pathogens, and for them, it's easy to have one. If you have one, if they say we will test for PRRS and they find PRRS, then they will change vaccination schedule or maybe the vaccine, so they know what to do. If you have, of course, 3 pathogens coming out, then you should look to the 3. And there is a kind of an idea, it's too complex. Why should we make it that complex? And that's a little fight at this moment, but this is temporary. Yeah. So I think it's a matter of a good education, and also not only during the university years of a vet, but also after they finish their studies. I think it's also up to us to give them a proper training how to deal with those things. And let's say the artificial intelligence and the translation will help, of course. If it's too complex, I can understand them that they say, we cannot deal with that. But if you help them in the translation of all the data, then I'm convinced that most of the people will use the new technologies.
Fascinating stuff. Hans, just one question coming in from our audience from Samuel. What steps should we take when we notice or have pathogens in our farms, considering the ban of antibiotics?
Okay, so yes, you should know that antibiotics are just, you're using it because you want to reduce the bacteria that are circulating there. Most of the time, the bacteria that are circulating, this is mainly due to the fact that there are other pathogens circulating. And I'm thinking in the direction, for instance, as PRRS, porcine reproductive and respiratory syndrome virus, or PCV2, porcine circovirus. These viruses are just dealing with the immune responses. So if you reduce the immune responses, then bacteria can come up. So if you know that you have those bacteria and you want to, deal with that with antibiotics, then this is not wise to only focus on the use of antibiotics. Then it's much better to go and have a look, and that's why this technology is so important. You should look what the other pathogens are that are circulating, and then I'm thinking mainly in the direction of certain viruses. So a better control of circoviruses, a better control of PRRS in a pig herd will, and everybody knows that, will reduce the use of antibiotics. And if you reduce the amount of antibiotics that are used, this will also help in controlling the resistance, of course, the whole story on the resistance. Also interesting is to tell that with PathoSense, that new technology, we are also able to look to the resistance. We have done that already with specific pathogens. At this moment, this is in full expansion. So in a longer term, we will not only identify bacteria, we will also find virulence factors, but we will also, based on the genetics of the bacteria, we will also be able to identify antibiotic resistance. So that means that everything will be done in one sample, and that will make the life, of course, of a vet much easier than before. But antibiotic resistance is a big story and is not only linked with bacteria. This is a much broader thing. Also, environment. If you have bad environments, bacteria will have a proper environment to replicate, to multiply. So everything is linked with each other, and that's why we should put efforts in combining all those things. But in the future, resistance will also be identified by just genetic analysis. Okay, Barry.
And thank you so much. I think we're just going to have to leave it now.
Okay.
It's just gone past 10 o'clock. And thank you very much for your excellent presentation and for
Thank you, Barry.
addressing all of those questions. Thank you very much, Hans.
Thank you,
So
Barry.
we'd just like to thank once again our excellent panelists this morning, Professor Anne Lespine and Professor Hans Nauwynck, for giving us wonderful and insightful presentations and providing us, I think, with great overviews of the respective projects this morning. The next Breakfast Club webinar will be in 4 weeks' time on Thursday, November the 20th, and I'll be sending out the invitations to all of you in the coming weeks. Again, just thank you very much to our panelists, and thank you everyone for attending and for your questions. We really hope you enjoyed this morning's webinar, and we hope to see you next time with us. Thank you very much, everybody, and have a great morning. Thank you so much.